First of all, I would like to thank Ben and Birgül, my co-organizers, to get together and just put our thoughts in this workshop. And as Ben defined described in the introduction, in the welcoming talk, we are just going to make something helpful for the students. And in this case, if you look at the program carefully, you will see that there will be some introductory lectures, then the case studies. So in this case, we are hoping that the students could learn better how the basic research methods and basic application of these research methods in environmental archaeology. Today, I would like to speak about the seeds and fruits, and my talk is about the basic research methods on how we are recovering these seeds and fruits from archaeological sites. But first of all, I would like to make a general definition of archaeobotany. Archaeobotany is the study of plant remains, obviously, that are recovered from archaeological sites. But more broadly, archaeobotany studies past human use of plants and the impacts upon the flora. It involves both the recovery of preserved plant remains from archaeological excavations and the application of botanical knowledge, including the identification of such remains to archaeological questions. So archaeobotany actually is like a larger framework for different subdisciplines or some specialization areas. These specialization areas are carpology and anthropology. Carpology is what I am doing basically. It is the study of seeds and fruits. Anthropology is the study of charcoal remains. Then these two subdisciplines is called, the remains that are coming from these two subdisciplines is called macro remains. So we are telling this macro because they can be easily seen by naked eye. But there are also some micro remains. These are the palynology, which is the pollen studies, and the phytolith analysis. So today, actually, I am very much happy that in all these three other subdisciplines, we are going to have really great talks, and I think I also will learn a lot about these other disciplines. But I want to focus on carpology basically today. There are some basic steps in research methods that we can follow from the field to the lab. So the first steps are data recovery methods, then sampling strategies. These are called, these can be grouped into data collection. Then, and these are done in the archaeological settlement most of the time. Then we carry our samples or process samples to our lab, and in the third step we have to identify and document these samples and plant remains. In the final stage is the data analysis. In this stage, we are just trying to find some meaningful patterns and connections inside our data set. So these are the four basic steps of archaeobotanical research, or any kind of research actually. So I would like to follow these basic steps. First of all, this is the, I would like to start with the data recovery methods. when you are in an archaeological site, and if you want to start an archaeobotanical project, the most important thing, of course, you must have some ideas about what kind of material you are going to deal. So in this case, you should know the preservation conditions, for example. Usually beforehand, before start the excavation, we can have some ideas about preservation conditions. Then you should also know the soil types and what kind of depositional conditions you are going to encounter during the excavation, because these aspects change the preservation condition and also other processing of the other processes in this data recovery. And finally, you should know that there should be some equipment, and your equipment should fit to your expectations. So this is also an important issue, and you just have to follow, you just have to find some solutions how you are going to process your samples, your soil samples. All right. I would like to start with preservation conditions, because there are many different preservation conditions around the world. But I will just describe three of them for our purpose here. The first one is the carbonization, and this is very common in the Near East and in Europe. Carbonization is a chemical process that lets the initial shape of the seed or fruits some a little bit deformed, or a certain aspect deformed, but as you can see in the pictures at the left side, there are different type of seed remains and fruit remains. So their overall shape is preserved by carbonization. Carbonization, this chemical process occurs between 200 from 200 to 400 degrees. So if you go over 400 degrees, the remains, the seeds or fruits, will be deformed too much, and it is getting too difficult for identification. However, for example, some plants produce fruits and seeds with hard shells. Yeah, like you can think that olive, for example, is it has a fleshy part that we eat, and also there's a hard shelled inner part where the seed is enveloped. So in some cases, in some cases, we can also find that these hard-shelled fruits and seeds. So it is important to know the basic plant anatomy, that that you can have less deformation because of carbonization in this different type of seeds and fruits. Here you can see that how carbonization changes the overall morphology of the seeds. This is a emmer grain, and you see that the grain is just puffed up when the temperature is getting higher. Another preservation condition is the waterlogging. It is common in Europe, near lakesides, peats, and marshlands where there are anaerobic conditions. This means that there are some microbe or bacteria-free conditions, and then by this way they can be these seeds and fruits can survive inside the water. So you can see at the left side that there are the basic there are there's a less morphological deformation in comparison to carbonization. At the left side, you can see different type of seeds, and even in some of them, you can see that their color is preserved. But Also, more importantly, very fragile plant parts like leaves, roots, tubers, and stems can also survive in this type of preservation condition. And in comparison to carbonization, or in comparison to the information that we can get from carbonization, we can find a more diverse set of plants in waterlogged conditions. Another preservation condition is desiccation. This is very common in very arid environments like southern Mesopotamia or Egypt. And in these pictures, you can also see that there is less morphological deformation than carbonization. And also, you can easily find the fragile plant parts which cannot survive carbonization. And these plant parts, as you can see at the left side, it can be like a fleshy part of garlic. Or at the right side, you can see the artichoke bracts, which is very difficult to get survives in carbonized conditions. But there are also many more places that we can find plant remains. For example, we can find them as in the left side and right. You can see that as imprints inside vessels. Or if you cut the vessel, you can find that there are different organic remains which were used as binding agents. And here at the right-hand side, you can see some dead bodies, and it's also possible to open the stomach of these preserved human bodies and look what they have eaten the last time before they die, for example, what they have got in their stomach. So, and there are many more. For example, there are many coprolite remains, you mean this human or animal feces. You can also find, we can also find some plant remains. Here, in this second part, we can maybe speak about depositional conditions. Here, you can see a very classical example of description how the deposition in archaeological settlements is working. In here, in the first stage, you can see a well. This is a Roman well. And you can see that it is still in use, and there is so little sedimentation, and because people are taking care of this feature, right? But when this feature is abandoned, these natural sedimentation processes starts, and you can, in this second picture in the middle, you can see that there is a type of sedimentation starts from either from here or like the well is started to fall down, for example, the walls over here. And the water table is going up somehow because of the inclusion of the other materials. Of course, the water is going up. Then there are some weed seeds or small animals can fall inside of this. So there's this natural sedimentation. But in the third stage, we see that the people of later times, they are just using this context as a rubbish dump. You know, they just put all kind of rubbish from their old settlements inside the well. So in this case, the sedimentation change, and there's an anthropogenic aspect of the sedimentation. This here in here you can see that there's this organic remains are just going down, of course, and then it seems that this part could be dumped. It will be better than the upper part, because as you know, there's the water table is getting higher over here, and now it's getting drier when you go upward in this context. So so when you are approaching to this kind of archaeological context, basically you have to know the history of the archaeological context. So it is very important that the archaeobotanists should be in the settlement. Here you can see a basic schematic representation of the different depositional environments and the survival chance of organic remains. Here, as I described you, this waterlogged environments is better than the drained or dry environments, basically. And here you can see that the number of remains or the diversity of remains is changing according to the acidity of the water. Here, as you can see, there are less water there are more archaeological organic remains, at least in basic calcareous sediments, instead of the acidic sediments, acidic water. I'm sorry, watery environments. So in dry, so this actually affects our data sets that in dry environments we can find less remains because they are not. They are not able to survive different natural agents impacts. So the third one in data recovery methods is the processing equipment. Basically, we are collecting or we are recovering our plant remains from with a system that we call flotation. This is a technique called flotation. Here in the left-hand side, you can see that these charred plant remains over here are just staying, going up because they are basically empty from inside. In that case, someone is holding the water bucket, and another person is scooping this out. This is a method that first developed during 60s, and Stuart Struwer, just one of the person who developed this method in 60s at Apple Creek site. But there are in time, after the first methods developed, there are also many different type of processing techniques appeared, but the basic understanding is the same. Basic system is the same. That here at the left side, this is Ankara flotation, which was devised by British Institute at Ankara scholars who are working there. There is stiraf flotation in the middle, you can see, and bucket flotation is, as you can see, this is the same system that I have shown before. So there are this kind of different flotation techniques. But as I said before, the soil types are important, and if your soil is too much silty, then it will possibly not dissolve with these earlier flotation methods. So this is an important part that anyone working as archaeobotanist should take care of, because the previous methods works very well for clayey soils, but if the silt ratio is too much in the soil, you have to apply to another technique, which is called washover technique. And there's a very good YouTube video on YouTube that you can just find it if you write washover technique. So this technique is just washing the sample over, basically. All right, so after we process this sample in our flotation tank, two different fractions, we are getting two different fractions. The first one is the light fraction, which is the part that we analyzed in the lab. Here we are just hanging them on those lines and to get completely dry, and then we open and pack it to be sent to the lab. In the second fraction is heavy fraction. These are the part of the sample that doesn't float to here. All right. So I think that this sample is just under this this water here on top of this mesh. Then we remove this mesh from here and we put under the sun to dry. Then later on we are just analyzing or just sorting out this sample, find more charcoals or find more small micro bones from small mammals or shells or maybe sometimes metal slags or little small artifacts as well. So this heavy fraction is also usually rich. Then the second important step in research method is deciding the most suitable sampling method to reach a reliable and representative data set. There are different type of sampling strategies, which then they have got pawns on. There are some advantages and disadvantages. One of it is the judgmental sampling. This is basically the most common sampling method in the Near East. In this case, the archaeologist is just collecting the samples for us, and then we process the samples. And archaeologists, of course, tend to collect the samples from clear contextual archaeological contextual locations. So in this case, in this type of sampling strategy, we can just have rich findings with concentration of plant remains with a clear contextual information. But in this case, less conspicuous context wouldn't be represented in your data set. Then you can lose some information. So there's systematic sampling. This is the uniform sampling grid that first you devise the uniform sampling grids and you to recognize the activity zones in your archaeological context. Then you collect the samples from every little grids, all right, every squares. So in this case, one of the disadvantages is that there could be too many samples if you collect too many subsample in this way from every grid, and this is a little bit this can be a little bit difficult to process all of these samples. Then there is random sampling. In this case, the basic advantage is that before you start of making this kind of sampling, random sampling, you have to know the archaeological feature first. Then you can say that the samples will be taken from the from this certain archaeological feature, but it will not be taken from other archaeological feature. So in the Near East, especially, there are too many preconditions for the usual archaeological excavation, so it is very difficult to implement this kind of random sampling. All right, but perhaps the best method would be that some kind, for the Near East especially, some kind merging systematic sampling with judgmental sampling. Here you can see an application of systematic sampling. So this archaeologist is just putting the grids, smaller grids, over here, and she will take samples from this part. Then judgmental sampling could also be applied together with systematic sampling. Then you can just take the samples from, for example, this is a vessel here, or this is a heart feature, this is another vessel, and this is another feature here. So archaeologists can take samples from every different features here. Then it will be better to merge some kind of systematic sampling and judgmental sampling together. And here, for example, archaeologists or any archaeologist could easily recognize that the soil somehow could be could look different in certain features, like there could be more axial lenses inside the hearts. Okay. Then this is an application of this method, and you can see that here this archaeological context is a granary, and it has like layers of very thick layers of organic remains over here in the bottom of this granary. So what the archaeologists and specialists did is that making some grid sampling. They are just make grids. They just divided the context into different grids, and then they collect the samples in every other square here, as you can see. And also they divided carpological samples and charcoal samples in this area differently. So what we have is that you can see here in these little squares here that there are different crop remains, and then their representation. You can see how many crop remains are existing. Here, this is a very interesting study. Here you can see that for example, bread wheat is represented at the like southern this part of the granary more. But the rest of the crop remains are represented at the northern corner of this corner of the context. So this is an application. The third step is the identification and documentation of plant remains. So now we are in the lab, and we are going to work in the lab. For an effective sorting, identification, and documentation of plant remains, we need certain tools. So these tools are laboratory equipment, a comparative collection, and identification manuals. Then, after we have this and we make our identification, we start to make the visual documentation of the plant remains. Okay, then we have got here, you can see that the specialist here is just dividing the samples into different fractions with different size of the sieves. This is important because different types of plants are sorted differently according to their sizes. So, for example, in the larger mesh size, you can usually collect the crop plants like this one. This is the barley. Or you can usually collect these big seeded wheat seeds. You can just collect some shell remains, or there could be some little bones as well. So this is the reason that the first thing that we do before we start analyzing the plant remains, we are just dividing into different fractions. Usually there could be 3 or 4 fractions, and then we pack them differently in different boxes or plastic bags. Then we start our identification or sorting out and identification of these different according to these different mesh sizes. All right. When we look under the microscope, this could be a low power microscope, and it is like it's a stereo microscope with the magnification of 10 times or 60 times. Then we look at this seed in different we are looking at different morphological feature of the seed, and also from different views. Here you can see this is the dorsal view, ventral view, and lateral view. And also you can look at in the cross section or transversal view. Then there are some identification protocols that we learned that during our studies. So we apply these identification courses, I'm sorry, identification protocols into our object. All right. Then we have got these different identification manuals that we can check if our identification is correct, or we can check if that we can have any idea about how we can have some ideas from these manuals, how to, which family or which genus or species is our object. Then, most importantly, we use modern comparative collections, and these are the modern seeds that we can take out from our drawer and check under the microscope and try to establish this morphological feature, try to compare the morphological feature to make the exact identification. Here, this object actually is important for me, so I was not able to identify this plant remains, but it is very common. I must say it's very common at our site. It's gingerly. All right, there are some important descriptive parameters. These are general shape, like spatial shape, which means that three-dimensional view. The size is important for us. Surface pattern and surface texture is important. The position and visibility of the hilum is important. Shape and the position of embryo is important. So when we are drawing an object, a plant remains, first we have to locate this according to some principles, right? So ventral side is always where the hilum is located. So we know that hilum is at the ventral side, then we draw the object according to in that principle. So the dorsal side is the side facing away from the hilum. So it's the opposite side could be the dorsal side. And also the cross section, this is easy. All right, afterwards, we just count all of these plant remains in different, at genus level or species level or sometimes family level. It's related to the identification clarity. You know that if you identified Amaranthus sp, this means that you were able to identify until genus level. However, here you can see if you identify Salsolo Kali, this means that you identified to the species level. So we just count them and we make this type of sheets. Then in the visual documentation, you can see that we usually, I mean, in older times, it was being drawn, but in recent times, we are just taking the pictures of the object, which we think that it is important to report in our studies. However, drawing is always very still better. Drawing is always better if it is done correctly. You can see this is a flax seed. And here you can see some holes. And this is important. And I think the illustrator had done a good job over here. That by showing these little holes. Because these bubbles are just oil bubbles. That this flax plant is, the seeds of the flax is very oil rich. So there are very rich oil inside the seed. Therefore, because of the carbonization, as you can see, this oil makes some bubbles here. And the illustrator show in two different views. As you can see, the one part, this is the lateral view actually. The lateral view, one part is very much better preserved and the other part is not good preserved. All right, and scanning electron microscopy is important. This can provide high quality pictures, useful to study the morphological features, and it's very helpful for further documentation for us. Okay, in the last step, we have this pattern searching in the database, the data analysis step. In this case, I wrote pattern searching in the dataset. But I will come back to this. All right. There are different type of analysis that we can do. Maybe for the sake of the time, actually, I can just skip this part a little bit. But we have got this semi-quantitative analysis, which includes ubiquity scores, proportions, fine density, and ratios. So there are different these are different types of analysis, and we can then you are just trying to understand your data set by using this different type of semi-quantitative analysis. Then there are quantitative analysis, which are important for us, because if your data set is too big, if there are too many different taxa in your data set, it will be very difficult to control and try, and it will be difficult to find patterns inside your data set. For that reason, we are mostly using a type of analysis called correspondence analysis, and this is the unconstrained ordination technique, which means that you are not putting any constraining agent inside your data set beforehand. So this constraining agents could be time, environment, or it could be different type of groupings, you know. That could be the independent variable which affects your data set. However, in correspondence analysis and principal component analysis, we don't use this constraining independent variables. Okay, in this picture, you can see that here there are different sites, and also there are different, here you can see there are 4 different sites and 6 different crops. So you can see that barley appears 55 times in site 1. So in this case, this correspondence analysis and principal component analysis, they are just clustering the variables according to their relative proximities, occurrences to each species. Here you can see that site 1 and site 2, there are lots of lentil and barley remains. In site 3, there are some grape remains, but in site 3, for example, there is much less wheat grains. So this is the basic logic is like that. This is another type. If you want to constrain your data set with independent variables, you can use redundancy analysis or detrended correspondence analysis. In this case, in this 2 type of ordination technique, you will get an independent variable. Here, in this plot at the right side, you can see that I basically constrained this according to the time, basically, and time periods. And these are the subphases you can see. Here, the early Bronze Age and Iron Age, they are just clustered in very different positions. But very interestingly, the first phase of Iron Age, for example, clustered much different than these both groups. So these 2 ordination techniques is also important because they can also give us the statistical significance of our hypothesis. They are also calculating this. They are doing this Monte Carlo permutation test. So we can just we can just have a significant test after this ordination techniques. All right. In conclusion, I described 4 basic steps from field to the lab. These are data recovery, sampling methods, identification and documentation, and data analysis. So all these steps are important because what you are doing during these steps would possibly affect the reliability and representativeness of the outcome of your data set. So if you make a mistake, if you just ignore one type of analysis or did nothing, so this is the process that can affect the outcome, the basic outcome that you want to reach. Okay, the data recovery and sampling methods have to be planned before the excavation starts. At least you can have some ideas about how you are going to recover your data and how you are going to make the sampling. And also the most important part is the recovery and sampling methods must be consistent across the studied site. So basically, if you start with, I don't know, random sampling at one time, in one season, then you turn into systematic sampling. So this could affect the outcome very much, all right? So this recovery and sampling methods should be consistent. Then identification and documentation must be carried out according to the accepted principles in the literature. Identification must be convincing for other colleagues. And finally, according to the research problems, data analysis step can be either exploratory, which means that you can just put your problem, your research problem in the first hand, and then you can start your research, or it could be descriptive, that you can just try to find the pattern inside your data set, and this could be a descriptive method. Okay, all right. Thank you very much. Again, this was my last slide.